Hello, I'm SA, your AI essayist. Please note that all content, audio and video in this presentation are AI generated. As AI can sometimes produce inaccuracies, I encourage you to verify any factual information presented. With that in mind, let's begin.
The mid-20th century presented a formidable frontier in medicine. Diseases that today yield to targeted therapies often remained intractable adversaries, their mechanisms poorly understood, their treatments largely empirical. The pursuit of new pharmaceuticals frequently resembled a grand, often serendipitous, exploration rather than a directed scientific endeavor. Laboratories were arenas where educated guesswork met painstaking experimentation with the hope that a fortunate discovery might alleviate suffering. This era, while yielding significant advancements, was fundamentally constrained by a limited understanding of the intricate biochemical dance within the human body and the molecular disruptions that precipitate illness. Into this environment stepped Gertrude Belle Elion, a figure whose intellectual rigor and innovative spirit would profoundly reshape the very methodology of drug discovery. Her journey was one of deliberate, systematic inquiry. She approached the complex challenges of human disease with a scientist's keen analytical mind, seeking to unravel the fundamental processes at play and to devise interventions that addressed the root causes rather than merely the symptoms. Elion recognized that true progress lay in understanding the intricate pathways of life at a molecular level, and in developing compounds that could precisely modulate these pathways to restore health. Her early career was marked by a deep commitment to this principle of rational design, a departure from prevailing paradigms. While many contemporaries relied on the hope of accidental discovery, Elion cultivated an environment where scientific understanding drove innovation. She possessed an innate curiosity, a relentless drive to comprehend the why behind biological phenomena, and the intellectual courage to forge new paths based on that understanding. This foundational approach set her apart, positioning her as an architect of a new era in pharmacology, one where the development of life-saving medicines became a science of precision, informed by deep respect for biological complexity. Her work would ultimately transform the way we combat a spectrum of diseases, offering hope and healing where previously there was often only grim prognosis. The impact of this shift, though often unseen by the public, resonates through countless lives saved and improved, a testament to her singular vision and unwavering dedication to scientific advancement. The mid-20th century presented a landscape of medical challenges where understanding the fundamental mechanisms of disease remained a frontier. Inherited metabolic disorders, conditions arising from errors in the body's biochemical machinery, represented a significant portion of these unsolved puzzles. Diseases like gout, characterized by the accumulation of uric acid and certain forms of leukemia, where blood cell production went awry, were often managed with treatments that offered symptomatic relief. The prevalent approach to drug discovery at the time frequently leaned on serendipity, a hopeful exploration of chemical compounds with the expectation that some might prove effective. This method, while yielding occasional breakthroughs, was inherently inefficient. Gertrude Elion, however, approached these complex biological problems with a fundamentally different mindset. Her early career was marked by a profound commitment to developing a rational, hypothesis-driven approach to drug design. This involved a meticulous effort to understand the precise biochemical pathways involved in disease states. She recognized that by comprehending the molecular underpinnings of an illness, one could then logically design molecules to interfere with those specific processes. This was a deliberate departure from the prevailing paradigms of drug discovery, moving towards scientific precision. Elion's intellectual curiosity was a driving force. She possessed an innate desire to comprehend the why behind biological phenomena, the intricate dance of molecules within living systems. This curiosity, coupled with a relentless drive for understanding, fueled her dedication to deciphering the metabolic anomalies that led to disease. She understood that diseases often stemmed from subtle yet critical disruptions in the body's complex network of chemical reactions. Identifying these specific points of failure was a crucial first step towards devising effective interventions. The prevailing scientific understanding of the time was beginning to illuminate the intricate details of metabolism, particularly the roles of nucleic acids and their precursors in cellular function and replication. Elion keenly observed these developments, recognizing their potential to unlock new avenues for therapeutic intervention. The challenge lay in translating this growing theoretical knowledge into practical treatments. Many diseases, especially those involving rapid cell proliferation or the accumulation of metabolic byproducts, were poorly understood at the molecular level, making targeted drug development a formidable task. Elion's early work was deeply rooted in this pursuit of mechanistic understanding. She focused on identifying the specific enzymes and metabolic pathways that were either overactive or deficient in disease states. The goal was to design compounds that could selectively modulate these pathways. For instance, in conditions involving the overproduction of certain metabolites, the aim was to find molecules that could inhibit the enzymes responsible for their synthesis. This required a deep dive into the existing literature, a thorough grasp of organic chemistry, and a keen ability to synthesize new chemical entities with specific biological activities. This commitment to a hypothesis-driven approach meant that Elion and her colleagues at Burroughs Wellcome, later GlaxoSmithKline, or designing molecules with a specific purpose based on a theoretical understanding of a biological target. This intellectual rigor was a hallmark of her scientific philosophy. She believed that true progress in medicine would come from understanding the fundamental processes of life and then devising precise interventions to correct aberrant functions. The scientific environment of the mid-20th century, while advancing rapidly, still presented significant hurdles. The tools for analyzing biochemical pathways were less sophisticated than today, and the sheer complexity of cellular metabolism was a daunting subject. Yet, Elion's ability to synthesize information from diverse fields, including biochemistry, organic chemistry, and pharmacology, allowed her to forge a path forward. She conceptualized drugs as active participants in biochemical processes designed to interact with specific molecular targets with a high degree of specificity. This focus on specificity was crucial. The challenge in treating many diseases, particularly those involving rapidly dividing cells like in certain cancers, was to inhibit the abnormal processes while preserving healthy cells. Elion's approach aimed to exploit the differences between diseased cells and healthy cells, or between normal metabolic pathways and those that had gone awry. This required an intimate knowledge of the biochemical signatures of disease. The diseases she initially focused on, such as gout and leukemia, provided fertile ground for this new approach. Gout, for example, involves the deposition of uric acid crystals in joints, a consequence of the body's inability to efficiently break down purines. Understanding the metabolic pathway of purine breakdown became paramount. Similarly, certain leukemias were understood to involve an abnormal proliferation of white blood cells, suggesting a disruption in the regulation of cell division and metabolism. Elion's early work laid the groundwork for addressing these complex conditions by targeting their underlying biochemical roots, a strategy that would come to define her illustrious career and revolutionize the practice of medicine. Her early explorations were about understanding the disease at its most fundamental level to engineer a solution. The biological machinery that governs cellular life, particularly the intricate processes of cell division and growth, presented a profound challenge in the mid-20th century. Cancer, characterized by uncontrolled and aberrant proliferation, stood as a formidable adversary. The fundamental problem lay in the inherent difficulty of distinguishing between healthy, rapidly dividing cells and malignant ones. A therapeutic approach that could selectively inhibit the growth of cancerous tissues while preserving the body's normal, active cells was urgently needed. This required a deep dive into the very building blocks of life, the nucleic acids, and the metabolic pathways that produce them. Gertrude Belle Elion's intellectual journey led her to recognize that cancer cells, in their aggressive pursuit of replication, often exhibited heightened dependencies on specific metabolic pathways. These pathways, particularly those responsible for synthesizing purines and pyrimidines, the essential components of deoxyribonucleic acid, or DNA, and ribonucleic acid, or RNA, offered a unique point of intervention. Cancer cells, with their insatiable demand for DNA and RNA to fuel their rapid division, often consumed these precursors at a far greater rate than normal cells. This differential metabolic activity provided the conceptual leverage Elion sought. Her groundbreaking strategy involved the design of molecules that acted as antimetabolites. These were compounds meticulously crafted to mimic the natural purine and pyrimidine bases that cells utilize to build DNA and RNA. By appearing as familiar substrates, these antimetabolites could infiltrate the metabolic pathways. Once incorporated or encountered by the cellular machinery, it would disrupt the normal synthesis or function of nucleic acids. The brilliance of this approach lay in its biochemical specificity. By targeting enzymes or processes uniquely critical to the accelerated metabolism of cancer cells, Elion aimed to achieve a therapeutic effect while preserving the integrity of healthy tissues. This pursuit demanded a sophisticated understanding of enzyme kinetics, substrate specificity, and the intricate feedback mechanisms that regulate cellular metabolism. Elion's laboratory at Burroughs Wellcome, now part of GlaxoSmithKline, became the hub for this pioneering work. She meticulously studied the existing knowledge of purine and pyrimidine biosynthesis, identifying key enzymes and intermediates. The goal was to design molecules that could either block essential steps in these synthetic pathways or be incorporated into nucleic acids in a way that rendered them non-functional, thereby halting DNA replication and cell division. One of the earliest and most significant triumphs arising from this strategy was the development of 6-mercaptopurine, or 6-MP. The scientific rationale behind 6-mercaptopurine was rooted in understanding the purine salvage pathway, a crucial route by which cells can reuse pre-existing purine bases. Elion and her colleagues recognized that cancer cells, with their high metabolic turnover, were particularly reliant on this pathway for rapid nucleotide synthesis. 6-mercaptopurine was designed as a thiopurine analog, meaning it resembled natural purines but contained a sulfur atom in place of an oxygen atom at a critical position. This cell structural alteration proved profound. Once inside a cell, 6-mercaptopurine is converted into its nucleotide form, 6-mercaptopurine ribonucleotide. This active metabolite acts as a potent inhibitor of several key enzymes involved in de novo purine synthesis, the pathway where purines are built from scratch. Crucially, it interferes with the conversion of inosine monophosphate, a vital precursor, into adenine and guanine nucleotides, the fundamental basis of deoxyribonucleic acid and ribonucleic acid. Furthermore, 6-mercaptopurine can be incorporated into deoxyribonucleic acid and ribonucleic acid, leading to chain termination or the production of faulty nucleic acids, both of which are lethal to rapidly dividing cells. The selective toxicity stemmed from the fact that cancer cells, with their accelerated nucleic acid synthesis, were more susceptible to this disruption. The administration of 6-mercaptopurine offered a targeted approach to interfering with the very engine of cancer proliferation. The conceptual framework established with 6-mercaptopurine extended to other therapeutic areas and laid the foundation for a new era of rational drug design. It moved medicine away from serendipitous discovery towards a more systematic, science-driven approach to creating medicines. By understanding the molecular basis of disease, specific biochemical targets could be identified and molecules designed to interact with those targets precisely. This was a paradigm shift, transforming the search for cures into a problem-solving endeavor grounded in biochemistry and pharmacology. The success of 6-mercaptopurine in treating certain types of leukemia underscored the power of antimetabolite therapy. It offered tangible hope for patients battling diseases that had previously presented few effective treatment options. The ability to design a molecule that could interfere with the fundamental processes of cell replication and to do so with a degree of selectivity represented a monumental leap forward. This work involved deciphering the biochemical language of disease and then writing a counter-narrative that could restore health. Elion's meticulous approach, her relentless pursuit of understanding at the molecular level, and her innovative application of biochemical principles to drug discovery set a standard that continues to influence pharmaceutical research today. The targeted disruption of purine and pyrimidine metabolism became a cornerstone of cancer chemotherapy, a testament to her profound insight and rigorous scientific methodology. Elion's scientific acumen extended beyond the realm of oncology, venturing into areas of metabolic disturbance and immune system regulation with equally groundbreaking results. One significant challenge faced by physicians and patients alike was gout, a painful condition characterized by the accumulation of uric acid crystals in the joints, leading to inflammation and severe discomfort. The underlying issue in gout is often a metabolic imbalance, specifically the overproduction or underexcretion of uric acid, a byproduct of purine metabolism. For decades, treatments focused primarily on alleviating the symptoms of gout attacks, offering temporary relief without addressing the root cause of elevated uric acid levels, known as hyperuricemia. The limitations of these palliative measures highlighted a critical need for a therapeutic agent that could modulate uric acid production at its source. Gertrude Belle Elion and her colleague George Hitchings embarked on the rational design of allopurinol. Within this context, their approach was deeply rooted in understanding the enzymatic pathways involved in purine synthesis and degradation. They recognized that xanthine oxidase, an enzyme crucial for the final steps of uric acid formation from hypoxanthine and xanthine, represented a prime target. The challenge was to find a compound that could effectively inhibit this enzyme, thereby preventing the excessive generation of uric acid. Drawing upon their extensive knowledge of purine analogs, they synthesized and tested numerous compounds, seeking one that possessed the right molecular architecture to bind to and block the active site of xanthine oxidase. Their meticulous work led to the development of allopurinol, a structural analog of hypoxanthine. Allopurinol functions as a suicide inhibitor of xanthine oxidase. Upon entering the body, it is metabolized by xanthine oxidase itself into oxypurinol. Both allopurinol and oxypurinol are potent inhibitors of the enzyme, effectively halting the conversion of hypoxanthine to xanthine and subsequently to uric acid. This biochemical intervention offered a profound shift in managing gout, moving from symptom management to addressing the underlying metabolic defect. Allopurinol's ability to lower serum and urinary uric acid levels dramatically reduced the frequency and severity of gout attacks and helped prevent the formation of tophi, the uric crystal deposits that can cause significant joint damage. The introduction of allopurinol revolutionized the treatment of hyperuricemia, providing a safe and effective long-term solution for millions suffering from gout and related conditions like uric acid nephropathy. The pursuit of understanding the body's biochemical processes also led Elion to tackle the complex challenge of the immune system's aberrant behavior in autoimmune diseases. In these conditions, the immune system, which normally defends the body against foreign invaders like bacteria and viruses, mistakenly attacks the body's own healthy tissues. This misdirected immune response can lead to chronic inflammation and progressive damage to organs and systems. The critical need was for a way to modulate or suppress this overactive immune response, enabling the body to remain protected from infection. The development of azathioprine emerged from this scientific imperative. Building on their success with antimetabolites in cancer therapy, Elion and Hitchings explored the potential of these compounds to suppress the proliferation of lymphocytes, the key cells involved in the immune response. They theorized that by interfering with nucleic acid synthesis, essential for rapidly dividing cells like activated lymphocytes, they could dampen the immune system's attack on the body. Azathioprine is a prodrug, meaning it is converted into an active form within the body. Once administered, it is metabolized into 6-mercaptopurine, the same compound that had shown efficacy in leukemia. The biochemical rationale was that 6-mercaptopurine would be incorporated into the deoxyribonucleic acid and ribonucleic acid of rapidly dividing immune cells. disrupting their function and proliferation. This targeted interference with lymphocyte replication offered a powerful mechanism for immunosuppression. The clinical application of azathioprine proved to be transformative, particularly in the field of organ transplantation. Following the advent of organ transplantation, a major hurdle remained the body's powerful immune response that would reject the foreign organ. Azathioprine, often used in conjunction with corticosteroids, became a cornerstone of immunosuppressive therapy, significantly increasing the success rates of organ transplants and allowing patients to live longer, healthier lives. Its application extended to a wide range of autoimmune diseases, including rheumatoid arthritis, lupus erythematosus, and Crohn's disease, where it helped to control the inflammatory processes and alleviate debilitating symptoms by tempering the immune system's aggressive actions. The development of azathioprine exemplified Elion's profound ability to translate fundamental biochemical knowledge into life-altering therapies, addressing conditions where the body's own defense mechanisms had become the source of disease. The mid-20th century presented a formidable challenge in the realm of the pervasive threat of viral infections. Unlike bacterial diseases, which could be effectively combated with antibiotics, viral pathogens proved remarkably elusive and difficult to target. Viruses, inherently simpler in structure and intimately entwined with host cellular machinery, resisted the development of specific therapeutic agents. The scarcity of effective treatments meant that infections caused by viruses like herpes simplex virus, varicella zoster virus, and Epstein-Barr virus often ran their course with little medical intervention, leaving a trail of discomfort, chronic conditions, and, in severe cases, life-threatening complications. The scientific community grappled with the fundamental question of how to disrupt viral replication while causing minimal harm to the host organism. Gertrude Belle Elion, ever attuned to unmet medical needs, recognized the profound impact a successful antiviral therapy could have. Her approach, deeply rooted in a meticulous understanding of cellular biochemistry, focused on identifying unique biochemical pathways or enzymes essential for viral replication, but absent or significantly different in healthy human cells. This pursuit of selective toxicity, a hallmark of her drug design philosophy, offered a promising avenue for developing antiviral agents. The challenge lay in pinpointing such a specific target and then devising a molecule that could precisely interact with it. Her groundbreaking work in this domain led to the development of acyclovir, a revolutionary antiviral medication that fundamentally altered the management of herpes virus infections. Acyclovir's elegance lies in its ingenious mechanism of action, a testament to Elion's profound grasp of viral and cellular metabolism. The key to acyclovir's efficacy resided in its selective activation by a viral enzyme, thymidine kinase, an enzyme produced by herpes simplex virus and varicella zoster virus, but present in human cells in a far less active form. Upon entering an infected cell, acyclovir, a guanosine analog, remained largely inert. However, the viral thymidine kinase possessed a remarkable affinity for this prodrug, phosphorylating it to acyclovir monophosphate. This initial phosphorylation step was crucial, as human cellular kinases were far less efficient at carrying out this conversion. Once monophosphorylated, acyclovir monophosphate became a substrate for host cell enzymes, which further phosphorylated it to acyclovir diphosphate and ultimately to acyclovir triphosphate. It is this acyclovir triphosphate that acted as a potent antiviral agent. It possessed a dual mechanism of inhibition against viral DNA synthesis. Acyclovir triphosphate competitively inhibited viral DNA polymerase, the enzyme responsible for replicating the viral genome. By binding to the active site of viral DNA polymerase, it blocked the incorporation of natural deoxyuanosine triphosphate, a vital building block for viral DNA. Acyclovir triphosphate also acted as a DNA chain terminator. When incorporated into the growing viral DNA strand, its unique structure, lacking the necessary 3' hydroxyl group for the addition of further nucleotides, brought DNA elongation to an abrupt halt. This effectively crippled the virus's ability to replicate its genetic material, thereby halting the infection. The specificity of this process was paramount. The requirement for viral thymidine kinase to initiate the activation cascade ensured that acyclovir was preferentially converted to its active form within virus-infected cells. This meant that healthy, uninfected cells lacking the viral enzyme experienced minimal conversion of acyclovir to its toxic triphosphate form. Consequently, acyclovir exhibited a remarkable safety profile with significantly lower toxicity to human cells compared to earlier antiviral agents or less targeted approaches. This selective activation was a masterstroke of rational drug design, embodying Elion's principle of targeting the unique vulnerabilities of pathogens. The impact of acyclovir on the treatment of herpes simplex virus infections, including genital herpes and herpes labialis, was profound. For the first time, patients could experience significant relief from the painful lesions and recurrent outbreaks associated with these conditions. Furthermore, acyclovir proved effective in managing varicella zoster virus infections, notably shingles, a painful reactivation of the chickenpox virus that can lead to long-term nerve pain. Its application extended to treating herpes simplex encephalitis, a rare, but severe brain infection. The success of acyclovir represented a paradigm shift in how antiviral therapies could be conceived and developed. The development of acyclovir demonstrated that by deeply understanding the molecular differences between viruses and their hosts, scientists could design drugs that were both highly effective and remarkably safe. Elion's meticulous research, her unwavering commitment to biochemical principles, and her ability to translate complex scientific knowledge into tangible therapeutic solutions were central to this achievement. Acyclovir stands as a powerful illustration of her visionary approach, offering a much-needed weapon against debilitating viral diseases and paving the way for future advancements in antiviral drug discovery. It solidified her reputation as a scientist capable of tackling some of medicine's most persistent challenges with ingenuity and profound insight. The successful development of acyclovir signaled an effective treatment for herpes simplex encephalitis and embodied a fundamental reorientation in the conceptualization and creation of antiviral therapies. This breakthrough underscored the power of elucidating the precise biochemical distinctions between viral pathogens and their human hosts, enabling the design of agents possessing both exceptional efficacy and a remarkable safety profile. Elion's rigorous scientific inquiry, her steadfast adherence to biochemical principles, and her capacity to translate intricate scientific understanding into concrete therapeutic outcomes were paramount to this accomplishment. Acyclovir serves as a potent testament to her forward-thinking methodology, providing a critical intervention against debilitating viral illnesses and establishing a precedent for subsequent progress in the field of antiviral drug discovery. It firmly cemented her standing as a scientist adept at confronting some of medicine's most intractable problems with both inventive acumen and profound perception. This systematic, mechanistic approach, so brilliantly exemplified by acyclovir, extended its influence far beyond the realm of virology, fundamentally reshaping the very architecture of drug discovery. Elion's work championed a transition from reliance on serendipity and empirical observation towards a deliberate, rational design process grounded in a deep comprehension of cellular biochemistry and disease pathways. Her philosophy centered on understanding the molecular underpinnings of disease, identifying specific targets that differed between healthy and diseased states, and then engineering molecules to interact with these targets precisely. This was a profound departure from earlier eras, where drug development often involved broad-spectrum agents or fortunate accidents. Elion demonstrated that a more predictive and targeted approach was not only feasible, but vastly more efficient and ultimately more successful in yielding safe and effective medicines. The implications of this methodological shift were far-reaching, laying the groundwork for what would later evolve into targeted therapies and personalized medicine. By focusing on the unique biochemical signatures of diseases, Elion's research provided a blueprint for developing treatments that could act with exquisite specificity, minimizing collateral damage to healthy cells. This precision was particularly critical in areas such as oncology, where the uncontrolled proliferation of cells presents a significant challenge. Her work in developing antimetabolites, for instance, which interfere with the synthesis of nucleic acids essential for cell division, offered a sophisticated means of inhibiting cancer cell growth. These agents, designed to exploit the heightened metabolic activity of rapidly dividing cancer cells, represented an early form of targeted intervention even before the advent of molecular biology provided the detailed genetic maps we possess today. Furthermore, Elion's investigations into purine and pyrimidine metabolism provided essential insights into the fundamental processes of cell growth and replication. Her ability to identify critical enzymes and metabolic pathways that could be modulated by therapeutic agents proved invaluable. For example, her research on azathioprine, an immunosuppressant used in organ transplantation and autoimmune diseases, stemmed from understanding how interfering with purine metabolism could dampen the immune response. This drug, by selectively affecting the proliferation of lymphocytes, demonstrated a sophisticated control over biological processes, a hallmark of Elion's scientific strategy. The success of such compounds validated her belief that a thorough understanding of cellular machinery could unlock novel therapeutic avenues. Her enduring legacy is intrinsically linked to her pioneering spirit in a scientific landscape that presented considerable obstacles, particularly for women. Navigating a field dominated by men, Elion's intellectual prowess and unwavering dedication allowed her to overcome societal limitations and establish herself as a leading figure. Her career trajectory serves as a powerful inspiration, demonstrating that scientific merit and groundbreaking discovery are universal. She consistently prioritized the scientific endeavor, often working with limited resources and facing skepticism, yet her commitment to uncovering the mechanisms of disease and developing effective treatments never wavered. This resilience and focus on the scientific mission itself are integral components of her lasting impact. The principles Elion established continue to inform contemporary drug discovery efforts, particularly in fields like virology and oncology. The development of highly specific antiviral drugs, such as those targeting HIV or hepatitis C, owes a significant debt to the foundational work she pioneered. These modern therapies often operate by inhibiting viral enzymes or blocking viral entry into cells, strategies that directly echo Elion's emphasis on targeting unique viral processes. In oncology, the burgeoning field of precision medicine, which tailors treatments based on the genetic profile of a patient's tumor, can trace its conceptual lineage back to Elion's early work in antimetabolites and her focus on exploiting biochemical differences. Her approach provided a crucial intellectual foundation for understanding how to intervene in disease at a molecular level. Elion's methodical approach fostered a culture of rigorous scientific investigation within the pharmaceutical industry and academia. Her research was characterized by meticulous planning, careful experimentation, and a deep commitment to understanding the underlying biological mechanisms. This emphasis on scientific rigor instilled a standard for drug development that prioritized thoroughness and evidence-based reasoning. It encouraged researchers to move beyond superficial observations and delve into the intricate details of biological systems, thereby accelerating the pace of discovery and improving the quality of new therapeutic agents. Her work provided a compelling argument for investing in fundamental research that explored the core processes of life and disease. In essence, Gertrude Belle Elion's contributions represent a profound evolution in medical science. She developed life-saving drugs and fundamentally altered the way scientists think about health and disease, offering a powerful framework for rational drug design. Her work transformed the pursuit of new medicines from a quest often reliant on chance into a systematic, science-driven enterprise. This paradigm shift has had a monumental impact, enabling the development of treatments for a vast array of conditions, and continuing to inspire new generations of scientists to tackle the most pressing health challenges facing humanity with intelligence, perseverance, and a deep respect for the complexities of life. Her influence is woven into the fabric of modern medicine, a testament to a career dedicated to alleviating suffering through profound scientific insight. Gertrude Belle Elion's legacy is that of an architect, one who meticulously designed the very blueprints for modern therapeutics. Her approach moved the quest for new medicines from a landscape often governed by serendipity to a meticulously reasoned, science-driven endeavor. She offered a potent framework for rational drug design, a methodology that has fundamentally reshaped how we understand and combat illness. This paradigm shift has yielded monumental benefits, facilitating the creation of treatments for an extensive spectrum of diseases. It continues to motivate new generations of researchers to confront humanity's most critical health concerns with intellectual acuity, unwavering resolve, and a profound appreciation for the intricate mechanisms of life. Her influence is deeply embedded within the very structure of contemporary medical practice, a powerful testament to a career devoted to mitigating human suffering through profound scientific understanding. The foundation for this transformative impact was laid by deeply ingrained conviction in the power of fundamental research. Elion championed an approach that prioritized understanding the core processes of life and disease. This commitment meant delving into the biochemical pathways and cellular mechanisms that underpin pathological conditions. She pursued a path of deep comprehension, believing that a thorough grasp of disease at its most elemental level would illuminate the most effective avenues for intervention. This perspective was instrumental in her ability to design molecules that could precisely target and modulate specific biological processes, offering therapies that were both effective and designed with an understanding of their interaction within the complex human system. Her work served as a powerful demonstration that investing in fundamental research was a direct pathway to tangible improvements in human health. The scientific community, and indeed the public, often grappled with a perceived distance between basic scientific inquiry and practical medical application. Elion's career provided compelling evidence that bridging this gap was essential for genuine progress. By meticulously dissecting the biochemical underpinnings of diseases like malaria, leukemia, and viral infections, she identified critical molecular targets. These targets became the focal points for her drug development efforts, transforming theoretical knowledge into life-altering treatments. This systematic approach, rooted in a deep appreciation for biological complexity, allowed for the creation of drugs with specific modes of action, minimizing unintended side effects and maximizing therapeutic efficacy. The elegance of her scientific philosophy lies in its inherent adaptability. While her early breakthroughs were rooted in understanding purine and pyrimidine metabolism, the principles she established proved universally applicable. The systematic identification of molecular targets, the rational design of inhibitory or modulatory molecules, and the rigorous testing of their biological effects became a repeatable model for tackling new diseases. As our understanding of genetics and molecular biology expanded, Elion's foundational principles provided the conceptual framework to integrate these new discoveries into drug development. The advent of genomics, for instance, offered an unprecedented ability to identify genes and proteins associated with disease. Elion's methodology offered the perfect toolkit to translate this genetic information into targeted therapeutic strategies, enabling the development of precision medicines that address the specific molecular signatures of an individual's illness. Consider the evolution of antiviral therapies. Early attempts to combat viral infections often involved broad-spectrum agents that could harm host cells as readily as they attacked viruses. Elion's work on acyclovir, a groundbreaking treatment for herpes simplex virus infections, exemplified her targeted approach. By understanding the viral thymidine kinase enzyme's unique role in activating the drug, she developed a compound that was selectively phosphorylated by this viral enzyme, thereby becoming active only within infected cells. This specificity dramatically reduced toxicity to healthy cells, a significant advancement that set a new standard for antiviral drug design. This principle of exploiting differences between viral and host cell machinery became a cornerstone for subsequent antiviral research, a direct lineage from Elion's pioneering insights. Similarly, her contributions to immunosuppression, particularly with azathioprine, revolutionized organ transplantation and the management of autoimmune diseases. The challenge in organ transplantation was to prevent the recipient's immune system from rejecting the transplanted organ, a complex process involving a cascade of cellular and molecular signals. Elion's development of azathioprine, an antimetabolite that interfered with deoxyribonucleic acid synthesis in rapidly dividing cells, including immune cells, provided a means to modulate this aggressive immune response. This allowed for successful transplantation of vital organs like kidneys, hearts, and livers, transforming previously fatal conditions into manageable chronic illnesses for countless individuals. The ability to carefully control the immune system, a delicate balancing act, was significantly advanced by her work, extending and improving the lives of millions. Her influence resides in the intellectual scaffolding she provided for future scientific exploration. Elion demonstrated that a deep, mechanistic understanding of disease could empower researchers to anticipate and design solutions. Her approach fostered a culture of inquiry that valued hypothesis-driven research and a meticulous, step-by-step process of scientific validation. This mindset is crucial in tackling complex, multifaceted diseases where simple solutions are elusive. The enduring relevance of her scientific philosophy is perhaps most evident in the current era of personalized medicine and targeted therapies. As we gain an ever more granular understanding of individual genetic variations and their impact on disease susceptibility and progression, the need for precisely tailored treatments becomes paramount. Elion's systematic, mechanistic approach provides the ideal framework for translating this intricate biological knowledge into effective interventions. Her work serves as a powerful reminder that the most impactful medical innovations often arise from a profound understanding of fundamental biological processes. Gertrude Belle Elion's career stands as an extraordinary testament to the power of intellectual curiosity, rigorous scientific inquiry, and a deep-seated commitment to improving human well-being. She synthesized knowledge, forging connections between disparate fields of biology and chemistry to create novel therapeutic strategies. Her systematic, mechanistic approach to drug discovery has fundamentally altered the landscape of medicine, enabling the development of life-saving treatments for a vast array of diseases. Her influence is woven into the fabric of modern medicine, a legacy of innovation and compassion that continues to inspire scientific advancement and alleviate suffering worldwide. She remains an exemplary figure, her intellectual rigor and visionary spirit a beacon for all who seek to understand and improve the human condition through the power of science.
We really appreciate you tuning in to this AI-powered presentation. Our goal is to push the boundaries of AI in content creation, and your input is invaluable. If there's a particular subject you'd like to see us tackle next, or if you have suggestions on how we could fine-tune our AI's creative prompts, please don't hesitate to leave your ideas in the comments section. We're excited to bring you more unique content very soon.